I'm excited to present this talk looking at whole brain connectivity changes under Midazolam during a pain and memory task. I'm Keith Vogt from the University of Pittsburgh. While you're reviewing these disclosures, I'll say this work is part of a broader program of research aimed at understanding how anesthetics affect cognition, particularly during pain. This abstract is a secondary analysis of data reported in a paper that was just recently accepted for publication that described both task activation and connectivity changes for two common anesthetics. And in that work, we had focused on a specific subset of brain areas known to be involved in memory, fear, and pain processing. The current work takes a broader discovery-based approach, employing a whole brain connectivity analysis, and we chose to focus on midazolam. 16 volunteer subjects participated in this portion of the experiment. They listened to a series of spoken words, and for each of them made a decision. For example, can it move or not? A third of the items were immediately followed by painful electric nerve stimulation. Thus, the entire experiment was really characterized by periodic painful stimulation. These procedures were done under saline and then repeated with different words under a steady state concentration of midazolam that achieved a light level of sedation where they could follow verbal commands throughout. We did functional MRI scanning during both segments, the goal being to compare connectivity between midazolam and saline. And it's important to point out, we first regressed out the timing of all of the task events to be able to compare background changes in connectivity. We used relatively broad regions of interest, or ROIs, defined by resting state data from the Human Connectome project. Areas in the brainstem and cerebellum were excluded, and this essentially constrained the analysis to a 30 by 30 matrix of region-to-region connectivity values. We can display the results of the connectivity graphically like this, and here you're looking under saline at mostly positive correlations in the time courses between those different regions within the brain. Then on the right, we see the same matrix of values under the midazolam condition. Taking the statistical difference between those two conditions gives this matrix of saline minus midazolam functional connectivity changes. There is an issue of multiple comparisons to consider, and these are connectivity changes that survive a threshold controlling the overall false detection rate at 5%. This matrix is also symmetric about that dotted line, and so we can remove the top half. What remains are independent connectivity changes between saline and midazolam. Based on the predominance of blue color, we can conclude that connectivity was greater under midazolam relative to saline. Data within these major boxes along the diagonal reflect functional connectivity changes between nodes within the same functional network, and we can see that these were less common. Looking at all of the off-diagonal boxes, we can see that cross-network increases in functional connectivity predominate, and an example is shown here between the sensory, motor, and default mode networks. This can also be viewed anatomically, and in this multi-view figure, where the width of the lines indicate the magnitude of functional connectivity change, we see bilateral distribution of functional connectivity increase under midazolam. And you can see there's a predominance of posterior changes compared to more anterior nodes. So midazolam increased connectivity in this study, which may or may not match your intuition, but consistent with its clinical use as an amnestic, long-term memory performance was decreased during next-day recognition testing. It's possible that the light plane of sedation that we achieved may have led to disinhibitory changes in the brain, and that could explain the increase in connectivity. It's also possible that afferent painful stimulation may differentially alter connectivity, and as expected, subjective pain ratings did not differ between the saline and midazolam periods. So it's an interesting and really open question as to whether these aversive experiences in this highly connected brain state might be implicitly encoded in a way that shapes future behavior despite the absence of explicit recollection. I want to thank FAIR for directly funding this study and recognize also my current support from the NIH. I welcome questions either asynchronously or during the live Q&A to follow.